My name's Ella. I'm a PhD student in the Department of Plant Sciences. So I work in a research group that's focused on C4 photosynthesis, which is a subtype of photosynthesis. It can be up to 50% more efficient than the default state, especially under conditions of high temperature and water limitation. And so my group is looking into how C4 photosynthesis evolved, what controls it, and how we can recreate characteristics of C4 in other crop species. It's really important that we are able to sustainably increase crop production to ensure food security for future generations. I'm working on a C4 species called Gynandropsis gynandra, which is a leafy vegetable plant that's native to various parts of Asia and Africa. C4 photosynthesis requires certain genes to be expressed in very specific cell types. So my research is trying to understand how, in the course of evolution, genes have been recruited from a broad role to a cell type specific role. So today I'm going to work on several different projects I have on the go. One of the things I'm doing is testing whether a certain transcription factor binds a certain DNA sequence to drive cell-specific gene expression. And to do that, I'm doing a bit of cloning, and I'm going to do kind of one step of the cloning process today, which is just to extract DNA from some E. coli cells that I've been growing and then get that DNA sequenced. And as well as that, I'm also going to plant some more Gynandropsis gynandra, which I germinated 2 days ago. I've been growing colonies of E. coli that contain the DNA sequence I'm after, and I took samples from these colonies, made them into liquid cultures, and then extracted the DNA from that, and that's what I'm putting into these tubes now to get sequenced to check that my bacteria have been growing the right DNA sequence for me. I'm just putting it in the Dropbox where they're going to pick it up and they're going to sequence it overnight, and I'll have the results tomorrow. I'm going to mix some soil now to plant these seeds in. These are the plants that I'm studying. I've just got some standard compost and some vermiculite. These are just fertilizer balls to help them along because they're quite fussy. And I'm just going to mix that with my hands and then put it into these trays and then put the seeds in and hope for the best. Basically, I've got 4 different genotypes here. Once I've grown them, I'm going to take leaf samples and look at how they differ in terms of their photosynthetic gene expression. Right now, we're inside the growth chamber. It's a controlled environment with, I think it's 22 degrees, consistent CO2 level. The lights go on and off in a regular photo period. Hopefully my plants will be happy in here. They're also safe from pests, which is a huge pro. In 5 weeks' time, these will be hopefully mature plants that I can take samples from.